Okay, so yeah, please begin, Mengliu.
Okay. Thanks for coming to my seminar today, everybody. My topic today is vasculature and airway extraction and analysis from 3D medical images. So, so the reason we choose this topic is because according to WHO 2016 statistics, the top 10 global causes of deaths, they are highly related to vasculatures and airway structures in the human body. So to facilitate diagnosis, it's very crucial to use automated analysis process using computer algorithm. Well, for there are a variety of anatomies, modalities, and pathologies with vasculature and airway data and medical data. For example, this is a slice of airway CT data, and these are two perspectives of intravascular ultrasound. These are two perspectives of the aorta CT with aneurysm pathology. And here is a slice of color fundus photography retinal data with AMD disease. We are not going to talk about every kinds of anatomy and modalities. And we are going to focus mainly on CT data in this thesis. And for the segmentation problem, we focus mainly on segmentation problem in this thesis. And for the mathematical formulation, there are three main formulation. One is the binary segmentation, which projects a 3D grayscale image eye onto a binary mask. So the output is either 0 or 1. And another formulation is to do tree tracking, which is an iterative process which accumulates data in the 3D image to form a tree structure. And the third formulation is to do bifurcation detection. So basically, the output result will be a set of 3D points which are not necessary to be continuous. So they can be everywhere in the image, which are the bifurcation locations. And in this thesis, we focus on the last two formulations, the tree tracking and bifurcation detection. The contribution of this thesis, we have 7 contributions. The first one is to use novel features for bifurcation detection, which was published in 2014 ISPI. And the second contribution is to use genetic algorithm search for parametric bifurcations in 3D images. We published it on CRV 2017. The third contribution is to use geometrical and topological tree statistics in locating trees, 3D trees in airway images, which we published in 2017 CRV as well. And then we progress on to using deep learning neural networks for tree tracking tasks. So we published another work on MICAI DLMLR 2018, which is the TreeNet, and it's the first deep learning network predicting tree directions. And then we had another paper using LSTM-based higher order Markovian tracking approach, which were published in 2019 MICAI MLMI. And then taking into the grant of global information, we used pictorial structure for tree detection and published this work in MICAI 2017. And then by equipping the attention module with LSTM and vasculature prior, we published another work in 2019 ICCV VRMI. So let's move on to the first contribution of this thesis, bifurcation detection in 3D vascular images using novel features and random forest. So the motivation is this. We want to locate all the bifurcation locations in a 3D tubular tree. And we try to use a machine learning method for doing that. So a bifurcation location, so for example, we can see here, a bifurcation is a place where a major branch bifurcates into 2 smaller branches, and we want to locate all those places in a 3D image. The contributions of this paper includes the following. we proposed novel features specially designed for 3D bifurcation detection using random forest. We proposed a generic tree tracking pipeline working with any kinds of 3D images. And we reached detection accuracy which can outperform state-of-the-art works. The pipeline is in the following. We take in a 3D patch image, we extract novel features as well as some common features, and we input those features into a random forest, and then we get a bifurcation prediction whether this 3D patch contain a bifurcation or not. And then we're going to encode this in a general tree tracking pipeline. We propose 3 novel features in this work. The first one is a multi-modal von Mises feature. So multi-modal von Mises feature distribution is, intuitively speaking, is more like a Gaussian distribution on a 3D sphere. So for example, on the lower left corner, we can see samples from a 3-modal VMF distribution. From 3-modal VMF distribution. So in our work, we use a multi-modal VMF distribution so to extract the multiple branches at a bifurcation location. The equation for the VMF distribution is in the equation like this, and for each p, x, theta I is a single model VMF distribution. We use the weight for the multi-modal distribution and encode it as the VMF feature. The second proposed feature is the histogram of eigenvector feature. So, for example, we use the eigenvector of a Hessian filter, and so, for example, we can see eigenvector field in the lower left figure here, and then we convert this eigenvector field into the corresponding histogram, and then we collect the counts in the histogram and turn into a COE feature. The third feature is concurrency of eigenvector. So basically, it's measuring the angles in the neighborhood, so to enforce the current point to be as close to the centerline of the blood vessel as possible. So it's here is an intuitive illustration of what is happening here. So we collect, so for each point in the center of the patch, we compare the direction we obtained at the current voxel and compare it to all the neighboring all the directions of the neighboring voxels, and then we generate the corresponding COE map. And we can see from the lower right figure here that the COE map actually has a higher value along the center line of the blood vessel, while it's relatively lower when it's going across the boundary. So it will enforce the prediction to be as close to the center of the blood vessel as possible. And then we propose a general algorithm for a tree tracking pipeline, which includes the initialization, the direction prediction, tracking a new point, and then we encode the bifurcation detection into the step to distinguish whether the current patch is a bifurcation or not. If it is, then we start detecting daughter branches, and then and then if the terminal point is not reached, we go back to the loop in the beginning, and if the terminal point is reached, then we stop the tracking. We work on 2 kinds of dataset. One is the Vasculature dataset, and one is a clinical dataset on Vasculature. For Vasculature, we collected 100 patches with single bifurcation and bifurcation free. And then for the Vasculature, we collected 105 patches with bifurcation and 86 patches bifurcation free. And here's a quantitative result comparing with a state-of-the-art tracking pipeline. So the red curve here shows the proposed method. The blue curves here show the competing method, and we're comparing the distance from the detected bifurcations to the ground truth and vice versa. And we can see a sharper curve shows a better performance of the proposed method. And here's another quantitative results comparing to another machine learning based method on for patch-based classification. So we can see the proposed method are the red bars here on binary classification accuracy. The blue bars is the competing method. So we can see a higher red bar, which means a higher classification accuracy of the proposed method. And here's some qualitative result. So the left one is the proposed one, and the right one is the competing method. And we can see the competing one has a lot of missing bifurcations. The second contribution is bifurcation localization in 3D images via evolutionary geometric deformable templates. And the motivation is this. So if we can describe the bifurcation as a parametric shape, how can we fit this model into the real image to find the bifurcation locations? And how can we find bifurcations simultaneously? The contributions of this paper includes the following. It's the first time we encode a parametric representation of 3D bifurcation. We designed multi-objective loss functions for this task, and we used genetic algorithm optimization with tribes for optimizing the loss function. So the shape model we built was on the left. It comprises 7 parts: the parent branch, the left and right transitional regions, the left and right curved regions, and the left and right straight regions. We use 7 excuse me. We basically fix the parameters for the shape, but we use 7 parameters for moving the shape. So we use 3 rotation parameters. We use 1 scaling parameter, and we use 3 translation parameters to move this model in the 3D space. We propose 5 different multi-objective fitness functions, which comprises 5 different functions, 5 different objective functions. So the first 2 are based on collecting vastness and intensity along the centerline of the model, and the next 2 are based on collecting the image gradients and comparing the image gradient and the model normal around the surface of the model. And the third one is to accumulate the intensity inside the whole volume of the model. So we add them up all together, and we get a multi-objective fitness function. And we're going to optimize this multi-objective fitness function to localize, to fit the model into a 3D image. However, this optimization task is a difficult task, so that's why we adopted the genetic algorithm to optimize this fitness function. The genetic algorithm basically has 2 major operations. One is to do crossover, and one is to do mutation. So, however, the genetic algorithm is used for detecting, for reaching the global optimization. However, in a real 3D case, we have to localize multiple bifurcations in the 3D image all at once. So that's why we use a tribes niching algorithm. So we form every bifurcation in the 3D image as a tribe. And by fitting each tribe into the image, and by fitting multiple tribes into the image, then we get multiple bifurcations detected all at once. So the tribes niching algorithm, the core idea is like this. The tribes should be limited within a certain distance, and if any population or any 3D model is above a certain distance from the center of the tribe, then we forbid this kind of either crossover or mutation. So that's how we form the tribe. We propose three different metrics for evaluating our result. The first one is the MD distance metric, which measures the distance, the Hausdorff distance between the set of the ground truth bifurcation and the detected bifurcation locations. The second one is the coherence metrics. We not only measure the distance between bifurcations, but we also measure the angle coherence between the parent branches and the daughter branches. And then we calculate the dispersion metrics. Because we have tribes here, so we want to measure how closely those populations are distributed into the tribe. So the more closely they distribute, that means the less false positives we will have in the final result. So that's how we design the dispersion metrics. We worked on two kinds of data set. One is Vasculature data and one is Vessel 12 data. For Vasculature, we chose 20 volumes with different Gaussian loss levels, and each volume contains 2 bifurcations. For Vessel 12 is clinical data set, and we chose 21 volumes containing 62 bifurcations in total. So this is a quantitative results on synthetic data, and we can see with different signal noise ratio, the proposed one outperforms the competing methods, the ESP7, the ESP14, and MBIA12 by have lower MC values, by having lower MP values, and also lower MD values. Not really. So basically, lower MC and lower MP values. And here is quantitative and qualitative results on clinical data. Again, we see that the proposed method have lower MC results and MP results, which show an advantage of the proposed method. And we can see from the left two figures here, the final results. So these are the detected bifurcations. And we can see that these the parametric model are pretty accurately converged to the real locations in the 3D clinical images. The third contribution, TreeNet, multi-loss deep learning network to predict branch direction for extracted 3D anatomical trees. So it was the first tree branch direction prediction by using deep learning method in this field. We proposed a novel multi-loss function with geometrical embedding, and the final performance outperformed state-of-the-art methods on clinical data. So the pipeline of this network is like this. So again, when we do tree tracking. So we extract a 3D patch along a certain point, and we take this 3D patch. So we accumulate all the training images like this, and we feed them into a adapted VNet, and then we by using a novel multi-loss function, we train this VNet, and then in the real inference stage, we will predict the tracking direction, which will be used in the tracking process. So the loss function we propose includes the following items. It includes comparing the directions on the of the 3D patch, and also it compares the mask on the cross-sectional plane of the the mask on the cross-sectional plane, and also the image intensity on the cross-sectional plane, as well as the estimated radius of the detected branch. So by adding all of them together, we form a novel multi-loss function, and then we optimize this multi-loss function to train our network. We evaluated on two data sets. One is synthetic data set, which comprises three categories to mimic real pathologies of clinical data. And we also evaluated on a exact online challenge data set, which comprises 7 anatomical structures. Here are quantitative results on synthetic data. So here we compared with 3 state-of-the-art model-based methods, OF, tensor, and German. And here are the 3 categories of the synthetic data: occlusion, torus, and leakage. So we can see that the compared state-of-the-art model-based work actually gave pretty low performance on the synthetic data. So we can infer that we're not saying that the models themselves are not correct. We're just trying. So we can infer that because these are model-based methods, so they don't scale very well to unpredicted pathologies like occlusion, torus, leakages, which are not classical tubular structures. So on the contrary, the proposed method scales very well on these kind of structures. And we did an ablation study, like throwing out different components of the loss function. And we can see that by adding all the components together, the multi-loss function reaches the maximal performance on direction prediction. And here is quantitative results on clinical data. So we compared with the particle filtering state-of-the-art method, and we compared on 7 anatomical branches in the airway. And here we are showing the error bars here. So the higher blue bars showing the larger errors of the computing methods, and they have larger mean errors as well as larger standard deviation. So which means the proposed method is more accurate and more stable in the direction prediction. And here are some quantitative results. Again, comparing the proposed methods to the 3 state-of-the-art model-based method on the clinical data. We can see that for all the branches and for all the levels of the data, the proposed methods outperform all the state-of-the-art works. Then the next contribution is tree LSTM, using LSTM to encode memory in anatomical tree prediction from 3D images. So the motivation is like this. The tree tracking process should retain memory along its path. It shouldn't. So the prediction in the past should have some kind of influence in the current prediction, and we're trying to encode that into a deep learning scenario. So the contribution of this work includes a formal derivation of the higher order Markovian process. We also propose a LSTM-equipped CNN network. We propose novel evaluation method for inspecting the correlation between bifurcation classification accuracy and sequential image evidence collected along branches. If you remember the tree tracking formulation we proposed in the very beginning of this talk, that the general tree tracking process is of lower order Markovian formulation. So basically, we start from a root point, which is x0, and by using a fixed tree tracking step, we augment from x0 to the next point, and then from the next point to the following point. So we can see the second line of the equation here. So every time we add a new point to that. So that's a lower order Markovian formulation. However, that's going to throw out the old memories during the tree tracking process. So we think, how can we encode a higher order Markovian formulation in this problem? We do this in this way. So we try to maximize the prediction at time step t for the prediction. So that's we're going to encode the prediction of time step t-1, t-2, t-3, t-4, etc., as well as encoding the evidence we collected along the tracking process. The evidence is it-1, it-2, etc. here. So we formulated into a higher order Markovian formulation, and we're trying to maximize the prediction here. However, in practical, this is intractable problem. So this is very hard to solve in practice. And that's why we are converting this higher order Markovian formulation into another formulation. So in our publication, we had like a formal derivation of the process, but I'm just showing the result here. So we derived the lower bound of this problems, and we are approximating the lower bound instead of the original problem. So by approximating the lower bound, we have to introduce some hidden variables. So we introduce the hidden variables to predict for the CT, and the hidden variable, and let me see. So we have to introduce the hidden variables here. There are different ways of estimating the hidden variables, but in this work, we use the LSTM to predict the hidden variables. So the whole pipeline is like this. We accumulate patches. We accumulate patches along the tree tracking process, and we use a pre-trained CNN module to generate the feature or the image evidence here. And we input these image feature into the LSTM module. So the LSTM have an automatic way of estimating the hidden variables, which it will use the hidden variables to predict the CT at time step t. So the green box here shows the hidden state estimation, and the red box here shows the output state inference. We apply this architecture on two different tasks. One is to do bifurcation detection. So we use a loss function for class prediction. And another task is to do direction prediction. So we have another loss for direction prediction. We evaluated our method on dataset exact09 on 3 major airway branches and 2 different tasks. So here is for bifurcation detection prediction. We use 2 baseline networks. One is the TreeNet and one is DCN for the image feature generation. And we stack LSTM on these 2 networks. When we compare to the baseline network, we can see that by adding LSTM, it actually increased the baseline performance by a lot on bifurcation prediction. And also here we can see on branch direction prediction, by adding the LSTM component on the original CNN-based network, the performance also increases. And then we ask, so how much does the result actually correlate with the evidence we collected along with the patch sequence? So because in the beginning we asked, in the beginning our motivation was the previous memory should have some contribution toward the current prediction. So we're asking how much those how much this information correlates with the prediction. So we did a correlation study and between the evidence and the prediction, the bifurcation prediction accuracy at the end of the sequence. And we can see that for TreeNet and for DCN, for all the 3 levels of the branches, we can see that the correlation is high. So that means that by using all the memories inside the patch sequence, it actually should help with the prediction. The next contribution is leveraging tree statistics for extracting anatomical trees from 3D medical images. So we have 3 contributions in this work. It's the first method that incorporates topological prior as well as geometrical statistics for anatomical trees into a tree tracking algorithm. We proposed a Bayesian-based tracking algorithm, and the final results has improved tracking accuracy and stability as well. So the method is like this. So we want to encode the dataset statistic into the real tree tracking process, and we try to break it down from 2 perspectives. One perspective is to encode the length statistics, and another perspective is to encode the angular statistics. For encoding the length statistic, we applied a Bayesian tracking process. So by using Bayes rule, we convert the length of the length of the branch into a into a weight which is measured by the cumulative distance. Anyway, so we can encode that into a Bayes formulation for the length statistics, and we also use the angular information into we also encode the mean and standard deviation of the angular information into the daughter branch detection. And we worked on 2 kinds of dataset. One is synthetic, which comprises 330 volumes containing 4 levels of trees. And the other one is the Exact09 challenge dataset of 7 branches of 3 levels. So, and here we can clearly see the statistics of the tree. So we extracted the mean and standard deviation of the length of the tree, as well as the mean and standard deviation of the angle of the tree, and we try to encode that. And for the clinical data set, we also extracted the mean length and standard deviation of the length, as well as the angles of the trees. So here are the quantitative results for the synthetic dataset as well as the clinical dataset. So we can see that the WOS means without statistics, WS means with statistics. We can see that with different noise levels across the synthetic dataset, with statistics hugely improved the number of branches detected. And also for the clinical dataset, by adding the statistics, we can also hugely improve the mean distance error and also the standard deviation. And here are some qualitative results during the real tracking process in the clinical data. So on the left, when the statistical information was not encoded, there could be, this is a possible case where the tracking process starts wrong at an early stage. And by encoding the statistic, we can see that the tree is labeled correctly during the tracking. The next contribution is the globally optimal anatomical tree extraction from 3D medical images using pictorial structure and minimal paths. So the contribution of this work includes we encode the geometrical and topological prior in tree extraction, and we also ensured a globally optimal tree extraction solution by using pictorial structure. So the pipeline is like this. Because it is a pictorial structure optimization problem, so it comprises two parts. One is the unary term, and one is the binary term. So we collected the unary term by using AN-based network to predict the tree likelihood and use it as a unary term. And then we collected statistics from the dataset and encoded it into the binary part of the pictorial model. And then by using the global optimization schema of the pictorial model, I'm sorry, I hear some background noise. Anyone have a question? Hello?
You're the only one that's unmuted, so.
Okay, great. So, so we use a pictorial model, and then we have, we convert, and so we use this pictorial model to extract bifurcation locations in the image, and then we use minimal paths algorithm to retrieve the centerlines of the tree, to form a whole tree. Yes, from mathematically speaking, this is the framework, the formulation of the pictorial structure. So yes, the unary term is, as I mentioned, is from the 3D patches and AN network, and the binary term is encoding like this. So we use the information of the covariance matrix we have collected from the dataset, and to retrieve the bifurcation locations. Well, we evaluated on two datasets. One is the synthetic dataset, and one is the Exact09 dataset. So we use three kinds of evaluation metrics. One is the ND positive numbers with tolerance ratio D, and M, which is the mean Hausdorff distance between the ground truth points to the detected set. And we also evaluated on branch extraction evaluation. So we measured the average distance between detected and ground truth center lines. Here is the result on synthetic dataset. We can see that how the M value changes along the different SNR. So as SNR grows smaller, the M value will grow larger, which is expected. And here are the results, quantitative results comparing to the state of the art. One checker and OF, which are all model-based methods. So the proposed one in ours, with or without statistics, with minimal paths. So by comparing on the M metrics, which is the Hausdorff distance, well, so by not using the statistics, we can see that the result is not as good. But by adding the statistic, we can see that the mean distance really gets smaller. And also, same thing happens to measuring on the MD, which is the mean distance between center lines. So by not using the statistics, it's better than the tracker method, but it's not outperforming OF method. However, by using statistics, we are outperforming. It's not really outperforming OF and minimal paths, but because OF and minimal paths, they were using manual bifurcation detections, and our method using automatic bifurcation detections. So our result kind of approached the state of the art, which using manual bifurcation detections. And here's another quantitative results comparing the state of the art, which is the checker method. So we can see our method has a higher the blue curve is sharper than the competing one, which means that the mean distance from the detected bifurcation to the ground truth is smaller to the competing method. The next contribution is the retinal image classification via vasculature-guided sequential attention. So in this work, we have three contributions. We leverage anatomical knowledge to guide attention maps, and we propose to use automatically extracted attention prior maps, and we encoded interdependency among attention features using LSTM. So the architecture is like this. The baseline architecture is a 5-level VGG16 architecture. So we input the original image, and these are different layers of the architecture. For each layer, we extract attention feature vector, and we input this attention feature vector into LSTM, and then we aggregate them together to make a final prediction. So and for the attention feature generation, it's like this. We extract the global feature from the last layer, and we unsample it, and then we combine this upsampled feature into the intermediate feature we extracted from the median layers, and then by combining the attention feature map into a prior image, we get the output attention feature. So the prior image, the prior image here are automatically generated by B-classifier filter, which is a common tool used for segmentation of blood vessels in retinal images. So we use two kinds of loss functions. One is the focal loss from the last layer, so to deal with class imbalance problem. And we also use dice loss from the attention map, comparing the attention map to the prior mask. And then we add them together. We add the dice loss across different layers into the focal loss, which form the total loss combined. And then we optimize the total loss. We evaluated on two different data sets. One is the age-related macular degeneration AMD dataset, which comprises 87 AMD diseases and 311 non-AMD diseases. We do a binary classification task. And another one is the diabetic retinopathy DB dataset, which comprises 413 training and 103 testing data with 5-level DB grading from non-parent to proliferate DR. And we can see the examples here. So for MD classification, which is binary classification, from this quantitative result, we can see that the proposed one outperforms the competing method, especially on the MD precision by at least 60% here. And we can see that even without the proposed network, by adding the vessel prior, it also outperforms the original network. So here is the 5-level retinopathy grading with different layers of attention. So for the first 3 layers, the first 3 rows, we use only 1 layer of attention, and for the next 3 rows, we use 2 layers of attention, etc., until we use 4 layers of attention. So we can see that by using 1 layer of attention, the accuracy and precision is not changing that much, but as we add more layers of attention, that we have a huge improvement in the accuracy as well as in the precision here. Well, as a summary, the thesis comprises 7 contributions. We use null features for bifurcation detection. We use genetic algorithm search for parametric bifurcation. We use geometrical as well as topological tree information in tree extraction, and we use deep neural network for prediction tree direction, as well as encoding higher order Markovian formulation. And then we use global optimal pictorial structure for tree detection. And then eventually we equipped this attention module with vasculature prior, which is encoded in LSTM. So as a summary of all the contributions, we designed generic tree tracking pipeline. We modeled appearance as well as geometrical side of the bifurcation. We encode statistical and anatomical information into the tree tracking process, and we propose to use semi-global as well as global optimization using genetic algorithm and pictorial structure. We utilized machine learning and deep learning for non-deterministic data-driven prediction. And we also encoded probabilistic searching approach by using Bayesian and Markovian formulation. And we combined different levels of analysis tasks such as tree extraction and disease classification. And we have this is a list of previous publications included in this thesis. And thank you very much. That's the end of